Very much, Chrissy. It's really my pleasure to be here and have this opportunity to share more around clinical data anonymization. It's a field that has really come in focus within the clinical development community over the last, I would say, 4 years, and there's an increased need for methods and increased needs for tools and skills to meet the new challenges of Policy 70 and soon Health Canada requirements around sharing of clinical data. So quickly about me, Christy has introduced myself. I've been working for 15 years in the life science industry. I've worked half of my career at Novo Nordisk and Ferring in Copenhagen, Denmark, as a statistician, programmer, and head of programming. And for the last 8 years, I've been working as an independent consultant on system implementation projects, clinical projects, submissions, but also for the last 4 years on data disclosure related project where I've been helping companies to implement data transparency initiatives. I've recently been appointed at the EMA Technical Anonymisation Group and also in the Health Canada Reference Group on public release of clinical information. Those two groups will provide in the coming, I would say, months to years, further guidance on how to anonymize data that are released through Policy 70 or through the new Health Canada policy related to the same topic. So I think we can expect this field to continue to evolve quite a lot over the next couple of years, and I think it's been really an exciting journey to be part of this, and I look forward to continuing progressing those topics. For today, I will tell you about some of the data sharing and identification guidelines that are available. I'm going to give you an introduction on residual risk assessment and talk more in details around the context of data sharing, the different contexts, and some insights into how to compute the probability of attack. And finally, I will tell you more about the fused data identification standard that was published in 2015 and for which I was a project lead, and use some examples to illustrate some of the challenges around data identification. So this webinar is based on a number of presentations I've given at industry events that I've tried to put together and update. So I apologize if some of the content is already familiar to some of you, but hopefully this will still give a good overview of what data identification requires. So this is a slide I really like that Pierre Vlastik from Sanofi presented some years ago in Amsterdam at the International Privacy Conference, which really represent the different concepts around anonymization and the different sharing circles that have different size and on which there are different controls. So when the data is collected, let's say at the healthcare professional, whether it is in the context of a trial or in the context of let's say going to the doctor or the clinic, the data is nominative. We want to be able to go back to the patients. We want to have their address, their phone number, being able to call them back. When the data is, let's say, passed on to the clinical development team, the data is pseudonymized. So all information such as name, address, telephone numbers, and so on are removed, but we still have patient IDs, date of birth, and all sorts of background information that will enable an attacker to identify patients with a very high probability. So the data that we receive in clinical development is not anonymized. It's still considered personal data and need to be treated as such. The data is often shared outside clinical development and could go to let's say other departments outside clinical development, which we will call R&D staff and partners, which typically don't receive the same training and don't have the same awareness around data privacy and personal data, and don't have the same systems on which there is security around the data. So at the moment, I think most companies still communicate data in a pseudonymized manner. to those partners. But we can see more and more companies who start looking at this circle as not external researcher, you could say internal researchers, where another level of anonymization may be required. When we go to external researchers, which could be an even wider community, there is much less control around the data. There's sometimes a data sharing agreement that is in place if the process is followed. There's a portal, but there is much less control in terms of what the researcher may do with the data. And here at this stage, most companies will anonymize the data. And of course, when the data go in the public domain, which is what's happening with Policy 70, the data needs to be fully anonymized. As we can see that as we go from one circle to the other, the sharing circle size potentially increase each time, but also the controls are lower and lower. And this is what justified the need for going from nominative to pseudonymized to anonymized data. So that's quite important to understand those concepts around controls, around sharing circles when talking about risk and anonymization. In terms of guidelines, this is just what was published in 2015, which was a very productive year when it comes to data identification guidelines. And they come from Europe, they come from US, Canada, and all those guidances or you could say mainly consistent within each other, but there's been quite many being written and updated since then. Recently, in 2017, a number of data protection agencies have also written their own guidelines. Some of them are quite extensive and quite technical. So the field continues to evolve extremely fast, and there's more and more awareness around data identification and need to anonymize data in many contexts. Data identification is a complex task. There's a number of processes around data identification in terms of handling the requests, in terms of the documentation that needs to be in place, how to process the data until it is anonymized, and so on and so forth. The exercise of identifying quasi-indirect identifiers within your data can be quite time-consuming. Applying the right rules to meet minimum data utilities is also very important. And finally, probably one of the most complex parts is to conduct a residual risk to ensure that the data can be shared and is below an acceptable threshold. And when we look at those guidances that were published in 2015, they were mostly covering, you could say, most of the areas, and hopefully they were overlapping quite a lot. At Fuse, we did put an effort in looking at one data model, which was CDSK-DTM, and really assess each variable one by one in terms of whether they're a quasi-direct identifier and what rule to apply. And in that context, we did work closely with Transferate, who was also developing their guidance at the same time to ensure at least consistency between the two of us. But again, they are consistent, but I would still recommend to read at least 2 or 3 of them to make sure that you really get potentially in some cases different opinions, but also that you get all the different areas of data identification covered before you start embarking on this journey. The disclosure process, so that's a slide that is quite simple, but I think is really straight to the point. We have to imagine that the lower side is, let's say, our clinical study, where we have a set of co-identifiers that potentially are anonymized. We will take date of birth and put into age, and we will have race and other information, and then we will have another set of information that is not necessarily in the public domain, that can be of high interest for the study, but that could also be sensitive information. On the other side, we have to imagine that we have a database of patients or people with name, addresses, so identifiers that can lead you, uniquely to persons, and also all sorts of co-identifiers, but not anonymized necessarily. And the disclosure process is really about being able to match those co-identifiers that are available, that can be available in the public domain or can be accessible through different means, and the ones that are anonymized in your study, in order to be able to link some sensitive information or other information that you don't know about someone in the first place to a given person. So hopefully this picture really explain clearly what's really at stake. Again, we don't have this information on the left-hand side in clinical data, but you know, nowadays it's quite easy to get co-identifiers data about people and find addresses and so on. It's really the other part that, in principle, should remain personal and should remain protected, that is potentially at stake in case of an attack. So Policy 70 is discussing the different types of possible adversaries and plausible attacks, and there are 4 of them that are mentioned. There's the first one, which is driven by financial interest, where an organization sees a financial interest in finding out who are the trial participants, and will usually require a strategy to actually identify a fair number of trial participants to make the search worthwhile. The second type of attack are demonstration attacks. So typically that would be a hacker or an academic who wants to make a name for himself, and he's just going to go and try to identify one patient to prove that company A hasn't done a proper job, or that they are very good at identifying people and potentially get something out of it. The third type of attacks would be the event in which an acquaintance examines a report and may spontaneously or actually not recognize someone that they know. So that would be a neighbor, a colleague, someone who knows that you've been in a clinical trial, and who will randomly or not go and look into those reports and find that person that they already have a number of information about that will enable them to recognize them. The fourth example that is mentioned by the EMA is around having a participant of special interest in the data. Typically, that would be a particular public person, a VIP, someone that has been focused in the press. But I think we can agree that this is unlikely to happen. Even though I'm saying that's the example I'm going to use afterwards to show some simple calculation. And this example of a participant of special interest, it can actually be extrapolated to other examples that are being talked about as plausible attacks right now in the industry. So the EMA doesn't tell you which of those attacks should be considered in your risk modeling. It lists some of them, and it tells you that the applicant should identify possible adversary and possible attack on the data and evaluate the impact on the risk of reidentification. At least they provide a path forward and a way of thinking in order to deal with this problem. The Policy 70 guidance, of course, talks about anonymization techniques. It does mention the Working Party 29 opinion and the 3 criteria of not being able to single out a patient, not being able to link to data points, not being able to do inference on a subject or a group of subjects, and does refer to Working Party 29 opinion 3 criteria as a technique that can be used, but also acknowledge that applying those 3 criteria will simply destroy completely your data. And the alternative that is put forward is to conduct a risk assessment. Right now, Policy 70 refer to a quantitative risk analysis or a qualitative one, where the studies will be considered as high risk, medium risk, or low risk, and the companies will define rules accordingly to deal with all the important co-identifiers. Transcelerate is providing a guidance around how to conduct the qualitative risk assessment, but it does feel that this qualitative risk assessment is in the policy right now during a grace period. The goal, I think, of the EMA is is to give enough time to companies to adapt and be able to conduct a quantitative risk assessment. But in the meantime, a qualitative risk assessment is accepted by the EMA. The question is for how long this grace period will last, but the guidance is still referencing those two approaches. So let's take this example just to show what data identification and risk computation can be. So remember the fourth example from for the plausible attacks would be to have a person of special interest. So let's take this example, even though it may be a stretch. So let's imagine that there is a study and there is someone that is very famous that is in the study, and this is talked about in the medias. And the data is hacked or is made available at some point in the public domain. So this is our VIP on the right-hand side. This is a presentation I gave in 2014 where he was about to marry a very young and very bright woman, and he was talked a lot in the press at the time. So I thought it was a good example to catch people's attention during my presentation. Anyway, he's still doing well, so I guess the slides are still up to date. So let's imagine that we have this pseudonymized data, typically what you would have in your clinical data that you collect for your clinical trial. We have patient IDs. We have not the most sophisticated ones, but let's imagine these are patient IDs: date of birth, age, gender, race, country. I did add partner age at the time because that did make him quite an outlier. But this is also co-identifiers in many ways. And if we had this data set and we knew that this VIP was in the study, which is again a very strong assumption, we could simply go in Wikipedia, look up his personal information. We will look at his birth date. We can even click and find more information about his wife, the second wife, and we will be able to for sure uniquely recognize this patient within our data. If we now do a bit of data identification, we remove the partner age, we remove date of birth that we put into age categories. Those categories are often seen and driven by some of the HIPAA requirements. Then all of a sudden, with the information we can find in the public domain about this person, there are three patients who can match the criteria. If we do more data identification and we remove age and we put age into age bands of 10, which is something that is done in many cases, and we now elevate country to continent. So United States now become North America. Now we have 5 patients that could be our person of interest. Now, if we delete all the data or all the demographics, of course we have to assume that there is all sorts of other information like we have in most of our clinical studies, but we know that this person is in the study randomly. There could still be, we could still find this patient with 1 out of 10 chance in the data. So this is just to illustrate that as we go through the process and we aggregate data more and more, there is more and more patients who become alike and make it more difficult to identify the person of interest. Which introduced the concept of occurrence classes. An occurrence class is a set of value for a selected set of co-identifiers. So in that case, these are the 6 co-identifiers that we have. We have to imagine that patient IDs have been recorded, and the occurrence class defined by this date of birth, age, male, white, United States, and partner age of 36 has a size of 1, which give us a probability of 100% to identify the patient, and so on and so forth. Now we have co-identifiers. We have 4 co-identifiers, and if we look at the occurrence class defined by age of 53, male, white, United States, we have an occurrence class of size 3, which give us a probability of identification of 33%. and so on and so forth. And here we get to 20%. So you have to imagine that we will do this for every single patient in the data, and we will assign a probability of reidentification, to every single patient based on which occurrence class they belong to and what is the size of the occurrence class. And this will lead to then computing some risk measures. And the two, you could say, most common ones that are listed in the literature is the average risk, which will be the average risk across all the patients. And then the second one is the maximum risk, which is 1 divided by the minimum occurrence class size. EMA policy recommend to use the maximum risk when it is a public data disclosure in order to make sure that every patient is protected adequately. The average risk is often used for control data disclosure. So typically what companies do when they share data with researcher with a portal and contract in place. What's possible also is to combine both and having a criteria on the average risk, but also having a less structured criteria on maximum risk to ensure as a minimum that, let's say, there is no unique patient in the study. But in the context of policy 70, this is really the maximum risk that is referenced and being used at the moment by sponsors. So before we move on, I just wanted to say that this example, even though is totally made up and may sound like a stretch, I think if we look at the assumptions again, the assumptions are okay beyond the fact he's a VIP. Is that the attacker has full knowledge about one patient and has also full knowledge about the study. So not only he knows all the, he has full knowledge about the patient co-identifiers, but he also knows exactly which study the patient has participated in, the sponsor, the study ID, and obviously the period and so on and so forth, which are very strong assumption. And this is, this example with the VIP that we are trying to identify is very similar to that other scenario where the attacker has full information about the patient and full information about the study. So in terms of the risk calculation, what we've seen is that it is possible to compute the risk just taking in consideration the clinical trial. So we're going to compute the occurrence class based on the clinical trial, and we're going to do an average or a max of both and compare that to a threshold. Another assumption that could be made is that the attacker, let's say, knows about the patient, that the patient has participated into a study, but he doesn't know which study. He knows that the study is an asthma study for company drug B, and it was about this period within those year or those 2 years. So he will have some, a certain level of information, but he won't have the complete information about which study. And then the number of study that falls under the range of possibilities is extended to what we call all similar sponsor clinical trials. And the term similar, I think, needs to be defined further. At the moment, we mainly look at obviously indication of therapeutic area, depending on whether it is applicable, the time frame on which the clinical study has taken place, and the sponsor, obviously. So you will compute your occurrence classes based on a larger circle where that your study belongs to, but you will still compute your average and your maximum based on a smaller circle. On the smaller circle, the occurrence class size or just larger. Another assumption would be that the attacker knows about the indication, knows about, let's say, the period, but doesn't know about the sponsor. He knows it's a drug in asthma, but he doesn't know which sponsor it is. He just knows that it is within this period and within this indication. And then you could say the spectrum of possibilities then extend to all similar clinical trials across sponsors, which then creates much larger occurrence classes. A fourth assumption would be to look at the entire disease population in the given geographical location where the studies is conducted, which I think for many people will be seen as being too liberal. But if you start considering this disease population, the new occurrence class would be a lot larger. And here it's not about making it up and taking the circle that will lead to the right reidentification score. Here it's really about doing a risk analysis and really see what's justifiable. Is it justifiable to consider that there is at least one patient that is at risk because someone knows, has full information about them, has full information about the study that's taking place? Is it fair to say that maybe the information about the study is maybe only restricted to the sponsor, and so on and so forth? So it's really that exercise that I think is fundamental for the companies to do when they compute risk calculation. Obviously, when you look at a small circle of let's say a study with few hundred patients, you start taking 5 or 6 co-identifiers, your occurrence class will be quite small. Will be quite small as a result. And it doesn't mean that it's wrong, but it means that many co-identifiers will need to be dropped or redacted. So I have a list of references at the end of the slide deck. There's one really good paper from Lucas Caniola from Biogen, who's explaining the concepts of population and how this has been implemented on their side, that I highly recommend you to read. So the probability of successful attack is a bit more than this. In the literature, what you will find in particular in the Institute of Medicine a report around data sharing, the probability of successful attack is looking at 3 types of attacks. The first one is when a researcher will attempt at identifying patients. And again, this assumes that we are sharing data via a portal and there are controls in place. But let's say a researcher, despite having signed a data sharing agreement, having access to a portal, will still try to identify patients. This is still something that is possible and even plausible, which will lead to the first type of attack. The second type of attack is the researcher or the data recipient who spontaneously recognize a patient from the dataset by simply browsing through the data. Even though there is no bad intention, this is still an attack. It's still considered an attack and needs to be managed. The third type of attack that we talk a lot is obviously the risk of breach. So typically, your organization hacks in the portal and retrieve the data. They can attack the data directly, or they can put it on the web, and anyone can download the data and attack the dataset. So those three types of attacks are different and can be managed through different factors. The attempt is managed through mitigating controls and also an analysis of motives and capacity of the data recipients. The acquaintance is managed, it's not managed, but it's actually computed to the study patient's prevalence. And finally, the breach is mainly managed by the security practice at the data recipient, but also in the case of sharing through a portal, the security around the portal. If you send the data to a researcher and you don't send it through a portal, it's mainly the security practice at the data recipient that is that will drive this probability. So it's important to evaluate the 3, which is the right-hand side of the equation. What is the probability of any of those events happening? And then knowing that those events have happened, what is the probability that there is then a reidentification of at least one patient? So before, what we've looked at was mainly the probability of identification, knowing that there's been an attack. And this left-hand side of the equation is managed through data identification, and this is where we can put our efforts as a data identifier. The right-hand side of the equation, which is the probability of attack, is mainly dependent on the disclosure context and what controls you've put in place, and about also who receive your data. So there are 2 ways to reduce your probability of successful attack. whether you put more controls or you do more data identification. What's important to understand that those two probabilities are obviously numbers between 0 and 1. So let's say that if your probability of attack if your 3 probability of attacks are below the threshold, the left-hand side of the equation can be equal to 1, and it doesn't matter really. And that's somehow what we do when we share data within clinical development. We don't conduct a risk assessment, but the probability of identification is equal to 1 on pseudonymized data. But all the controls in place, the fact that the staff is trained, the staff are hired by the company or contracted and could lose their reputation or could have huge consequences if they would do anything stupid with the data, does push this probability of attack to a very, to a level that is lower than an acceptable threshold. And of course, if there is a public disclosure, we simply assume that there will be someone who will attack the data and there will be a breach, and then the probability of breach is equal to 1, and then the rest really doesn't matter anymore. The only thing you can do is to work on your identification to bring the left-hand side of the equation below the threshold. So, and that's something that, if you go through the exercise and you document it well, will help you justify the data identification and rules that you've applied. So that was it for the first part of the presentation. The second part of the presentation will be focusing on the few identification standards, where I'm going to introduce you what we've done and why we've done that, but also I will take 4 or 5 specific examples that hopefully will illustrate better what is data identification in practice. So so let's get started. So it was back in 2014. We could see that especially the EMA was putting quite a lot of efforts to see how individual patient data from clinical trials could be shared more widely. And this is before Policy 70 was split between phase 1 and phase 2. At the same time, what we could see is that many sponsors were starting their own data sharing initiatives through portals, through putting a research request process in place, and so on and so forth. GSK and Roche started the whole process, I think, back in 2013 at the time. And then I think 20 to 30 companies have followed since then. And what we realize is that the data is coming in different data models, which is expected. I mean, some of the sponsors go back in time to 10, 20, sometimes 30 years to get data to researchers. That's at the time where, especially these data models were not widely used in the industry. Each company seemed to be defining their own high-level guidelines for data identification. Many of the companies have actually published them, which is really interesting, I think, for the community. But what was even more striking is that as a researcher, it's possible to request data from different companies within the same research proposal, which is, of course, a very positive thing. But as a researcher, you may not only get data from different data models, but you may also get data that are identified differently across sponsors, which may make the analysis more difficult and even error prone. So we felt there was a need to provide a standard that could be used to try to harmonize both how data is identified, but also to save time to companies who are making this data available. So we decided to focus again on CD's data models to have something extremely concrete. We gathered more than 20 participants at the time across the industry. We started to focus on SETM, and we looked at data privacy, meaning what are the quasi-indirect identifiers in the data, what rules and rationale to apply to identify them, and finally try to assess the impact on data utility that those rules will entail. So again, the goal is really to facilitate the assessment of direct identifiers, hopefully to help companies to save time doing that, ensure consistency in the identified data shared across sponsors, and also we've been providing some guidance on handling of low frequency and residual risk assessment in different data release contexts which is the appendix 2 of the standard that I also encourage you to read. I think the particularity of our group is that we are gathering people from across the industry and beyond. We have a number of data sharing specialists who are people working in pharmaceutical companies who are working day to day with receiving requests and identifying data and making sure the data is at the best quality possible for the researchers. We have CDISC experts that help us to understand these data models, but also the flavors in which they are implemented across the industry. We have data privacy experts, CROs who are conducting this type of work for many sponsors as well, software vendors who are developing solutions, and last but not the least, we have researchers in the group that help us understand what they do with the data, but also how our rules impact data utility and the analysis that they may want to carry on. That's just some pictures from some of the meetings we had over the years. So before we go forward, just a couple of disclaimers. So we're trying to provide standards based on the consensus of the working group. So the views do not represent the companies who are, you could say, providing experts in the working group. It's not representing some of the views of the experts in the working group either. In some cases, it's really consensus based. Most of the time, we manage to list pros and cons and agree. In some cases, it's more difficult. But when we've defined that standard, we decided to really, force ourselves to not try to reflect every flavor that is available in the industry in terms of the identification, but be able to link one given rule to one given scenario to really maximize the chance of having consistency in how the data is identified. Also, the rules that are described do not guarantee an acceptable or very small radial risk of identification. We can't just simply take the rules and the co-identifiers apply them and then consider the data is ready to be shared. We do recommend to companies to also look at the residual risk and do extra checks that will ensure that the data is adequate to be shared. So the key principles in our work was really to again differentiate direct and co-identifiers. We talked about that before. Direct identifiers will uniquely identify an individual, and the co-identifiers in connection with others will identify an individual with a high probability. So really this concept of uniquely identify or identify with a high probability is really important to understand when doing the assessment. For every co-identifiers and direct identifiers, we've been suggesting a primary rule and an alternative rule. The primary rule is more suited for proactive data identification, where you don't know what the request is about and you will try to maximize data utility. The alternative rule is often more suited for reactive data identification and special cases. We also tried again to assess the impact on data utility and also provide some implementation guidance when required. And last but not the least, we had to make some decisions sometimes that were not necessarily in line with what company A or company B were recommending, and we've tried to explain the rationales of certain assessments to make sure that the reader can understand the decision and overrule it or be convinced that this is the way to go. So we don't expect companies to implement necessarily 100% of the standard. But what we've seen around us is that many companies have been able to take it and at least implement 80-90% of it, and maybe customize some of it to their needs or their opinion, which is perfectly fine. We've been looking at different areas, and we've been defining rules for dates, low frequency and rare events, recording of unique identifiers, handling of free text variables and extensible code lists, geographical location, sensitive data. I'm going to talk about that a bit more later on. and so on and so forth. So it's quite extensive, you could say, the areas and the variables that we've been assessing, and hopefully this will help companies not to miss out on certain co-identifiers that may be hidden or may be not that obvious. And I will also take some examples of things that we've listed in the standard that we've simply gathered through all the experiences that were shared through the working groups from people actually doing data identification. Things that you will not figure it out right away when starting the first time. So if we take the example of dates, dates are also listed in EMA policy 70 guidance as an important co-identifiers to manage. In SDTM, dates come as relative dates, so typically study day, or they come as core dates, let's say 14th of July 2014, and those dates need to be managed. If it's a relative date, we consider it's already de-identified and no further identification is required. If it's a core date, we need to offset it. Special attention needs to be taken for partial dates, date of birth, date of death, or particular type of dates that needs to be considered separately. The case of adaptive design, where a patient enters a study into a new arm later on, also need to be looked at. Extension studies can create problems as well between the main and the extension part when doing offsetting. So we're also discussing date offsetting in more details across specific cases. We also list exceptions such as what's happening with medical history dates and patients over 89. That's something that actually came from the transfer rate guidance through our reviews that we've also recommended, where some people in the community realize that when patients are above 89, we often put them into that specific category due to the frequency in general, and this is what's recommended in HIPAA. But the problem is when you look at medical history dates and if those patients had an event close to birth, you may as well give away some more granular information about age. So that's typically the kind of things that I think very few of us thought about at the time when we started, and that were brought to our attention that we've listed in the standard. In terms of date offsetting, this is the algorithm that we recommend to use. It's a very simple and efficient algorithm. There are other algorithms that are probably as good, but the idea is to look at all your dates within the study period when the trial was initiated, which is, you could say, the first date when a date could be collected, and when the trial is completed, which is the last date when data could be collected. Obviously, there is medical history that is collected before those dates, but for each patient, the idea is to look at when they had their first visit or when the first piece of data was collected, and look at the distance with the trial initiation date, which is public, together with the completion date on repositories and registries like Udacity or clinicaltrial.gov. So the idea is to look at the delta back to the trial initiation date, and then apply this delta to every single visit, every single event, and do that for every patient. So every patient will have a separate delta based on when they've entered the study. And the advantage of this algorithm is that once you've applied it, all the visits, especially the first visits, are fully aligned. So all the patients have the same date of start. If they follow the visits as per the schedule, then their visit 1, visit 2, visit 3, and so on are also happening at very similar time, while the distances and you could say the sequence of events is preserved. And policy 70 guidance also recommend to apply algorithm that keep your dates within the plausible range of dates that have taken place in the study. As I said before, those two dates are public. If you start having a date outside this range in your data, then it is obviously offsetted, and yeah, this is something that would be obvious in terms of what has been done. While if it remains within the range, you keep some uncertainty in terms of what has actually happened, and it keeps your patients quite aligned and difficult to differentiate at the same time. We'll have to go quickly on those slides because there's 10 minutes left. This is an example on the issue with partial dates where we apply offsetting with a delta. So here in this example, we have an event taking place between visit 2 and visit 3. It's not clear if this event is before or after visit 2, but it's very clear this event is before visit 3, even though this date is partial. The algorithm tells us to apply minus 14 days in terms of delta, and after we've imputed offset and then make partial again, the visit 2 seems to be before event X, but event X now is not, we're not sure whether it happens before or after visit 3. So the sequence is not preserved, even though there was as well some uncertainty and some sequence here. So this is the kind of thing that we also point out in the deliverable to make sure that researchers are also aware that partial dates who are already unreliable when we have them in the clinical data or even more unreliable after they've been offsetted. The case of free text is also interesting. If we have a dictionary coded variable that is required or expected that is matching a free text variable, then we usually advise to remove it. If there is a dictionary coded variable that is permissible and let's say that is not that hasn't been completed or populated, then we will, as a primary rule, advise to review and redact the information. But if the variable is not important or not used for analysis, then it can simply be removed, obviously. Geographical location, what we find in clinical studies are obviously information about countries, about sites. Investigators also will give away the site and some geographical information. And for country, as a primary rule, we actually suggest to elevate the continent, mainly because it brings the risk lower by default as soon as you start enlarging the geographical location, but otherwise we recommend to keep it if it's important to analysis. If it's been requested by your in the research request as let's say a factor in your analysis, then you may need to find ways to compromise on other parameters in order to find country. Same for site and investigator ID. If they are not important, then we simply recommend to remove them. The interesting thing with geographical location is that other variables that seems unrelated could give away geographical location. For example, in the concomitant medication domain, the drug name can be different across countries. Companies, sponsors market drugs differently, sometimes across countries. So you may have elevated country to continent. It may be possible, if you haven't managed this variable, to find out what the country is. Race is an interesting one because in some countries you cannot collect race, like my home country, for instance, where, for example, you would elevate country to continent, but then the patients with a race missing may actually be the one from France, so you will not be protecting those patients very well when it comes to geographical location. Care of third party and labs could also be used in that context. So just to give you an idea again of some of those exceptions that are really not obvious that we've put into the document, that helps people to think about the data in a different way. Sensitive diagnosis, I think then I will be done. Sensitive diagnosis is a difficult one because some companies will consider that they are not re-identifying, and therefore all the management of other variables should ensure that patients are not re-identified to a certain extent. But other companies will say, well, in case the data is hacked, we will simply want to destroy any sensitive information, so patients cannot be harmed as a result, just as a safety measure. But when you take the case of sensitive diagnosis, there are sensitive data such as abortions, data about suicide that are easy to find in data. But if you take the example of HIV, which I've tried to do with the clients, we've tried to remove HIV information from a study that was a non-HIV study. There was a certain number of HIV patients, and if you look at adverse events, if you look at medical history, there will be a date of infection. There are certain drugs that HIV patients take that will identify them as having HIV, and of course there are certain lab parameters that have very high or very low values for HIV patients. So if you really want to remove sensitive diagnosis from your data, you would definitely need to understand the diagnosis, but you will also need to really pay attention to all the domains where this diagnosis could be indirectly hidden. So the deliverable, the standard is coming in the form of a spreadsheet plus some guidance documents that can be downloaded. It covers all the areas we discussed before, and there's about 1,300 variables that we've assessed. It's been downloaded about 700 times so far, and we've heard about a number of companies and academics who are using it and have used it as a reference for their data identification processes and implementation. The EMA guidance is also referencing the FUSE standard, mainly for the assessment of quasi-indirect identifiers, even though the guidance is related to phase 1 of the policy and unstructured data, the same concepts can be reapplied and reused in order to make the assessment. So in conclusions, well, there are many different data identification standards that are available. Again, there's a lot of consistency between them. People speak to each other, fortunately, and review each other's work. But I would really advise you to read at least 2, 3, or 4 of them to get a full picture. The identification of direct and co-identifiers requires a detailed understanding of study data and structure. There is some automation you can do to recognize your direct and co-identifiers, but it's quite important to read the protocol, read the CRFs, also make yourself familiar with the CSR to really get a better feel of what data is available in your study, if you haven't been part of the study, obviously. The analysis of data sharing context and possible attackers is key to cognitive risk assessment. Depending on the assumptions you make for the same study, the risk can be completely different. And finally, data utility is key and must be considered for both research requests and public disclosure. For the second one, for public disclosure, I don't think it's been clearly defined so far. Companies do their best to provide data that is useful, but the concept of data utility in public disclosure hasn't been defined clearly. The EMA has just celebrated their 1 year into Policy 70. I think they've shared these slides in the industry, and as you can see, there's been a lot that has been going on, and I think companies have put a great effort to make millions of pages available, and I think we can upload them all for all the hard work they've put on, and so far we can see from the data shown by the EMA that the initiative is rather well received. So well done to everyone who's involved as a researcher or as a sponsor or as a CRO or consultant. I think things are already going in the right direction. Before I close my presentation, some recommended readings that you can find. There is the FUSE data identification standard, and then there's a number of publications that we've done throughout the FUSE network that are available in the FUSE archive. So thank you very much. I don't know if there's time for a question or if I've been speaking for too long, Christian.
Thank you. No, thank you, Jean-Marc. That was fantastic.